pharmaceutical manufacturing, and process analytical technology, quality by design, and some of the key terminologies that are used in quality by design. Briefly, risk assessment, because it's one of the tools which is very important in developing products using QBD. And we look at applications of quality by design, in the area of product development, both new drugs as well as generic products, and analytical method development. Okay, see, if you see the current state of pharmaceutical manufacturing, conventionally, we have a batch process. Okay, we don't have continuous manufacturing as of now in the pharmaceutical industry. And quality assurance of the product is ensured by end product testing. That is, after the product is manufactured, you draw samples, test it, and see whether it meets the specifications. And there's always regulatory uncertainty, which is often cited by the manufacturers in their hesitancy to change anything, in the sense to introduce any new techniques. They are hesitant because they will be always worried whether the regulatory agencies will accept it or not. Okay, that's one of the problems which industry usually cites. But the significant opportunities exist for improving pharmaceutical development, manufacturing, quality assurance through innovation in the areas of product development, process analysis, process control, and automation. So if you see top reasons for recall of products in the pharmaceutical sector, you'll find that some of them are due to CGMP deviations, impurities and degradation products in the finished formulations, failure to meet USP dissolution test, presence of foreign substances, and some, very rarely, of course, marketed without an approved NDA or ANDA, and we have defective container being used. Okay, and in respect of packaging also, there are many issues like illegible labeling, incorrect or missing packaging insert. Either you have super potent product being packed in case of single ingredient products, recalls due to miscalibrated or defective devices, and sub-potent products in case of multiple ingredient, that is fixed dose combinations, and also super potent products in case of fixed dose combinations. So these are some of the different reasons for product recall. When it comes to product development, the current state is in most of the organizations, okay, and in the developed countries prior to introduction of ICHQ8, that is quality by design guideline. Okay, so the current state is method of development is empirical. Okay, you have trial and error experiments, and existing product knowledge comes in handy to develop a new product. And for generic manufacturers, you look at the innovator product information and try to prepare, develop your own product. And its initial trial batch is used for early assessment of how to go about the development. So what happens in empirical development is, usually, you just make one trial batch of a product. If it meets all the parameters which you have set, to put it for stability at, say 45 degrees. It used to be 45 degrees. Of course, now we do at 40 and 37. So if it, at the end of 1 month, if the product meets all the requirements and there's no visible, impurities or degradation or reduction in assay potency, you consider that as a best recipe, and go ahead, make 3 more batches of the same with the same formula and same process, put that for stability. Okay, and once you have stability data, you're done. You feel that you have developed a new product and you go ahead and launch the product. Okay, that's how it happens in empirical method development, which is followed currently by most of the generic manufacturers. Of course, discovery-based research organizations have now moved to the quality by design concept, which we'll be discussing in detail further. But what is the desired state of pharmaceutical manufacturing? See, what we discussed earlier was the current status. What should be the desired state? So desired state is product quality and performance is ensured through development of effective and efficient manufacturing processes, product and process specifications based on mechanistic understanding of how formulation and process factors affect the performance, and continuous real-time quality control and assurance rather than end product testing. Okay, there's a desired state. What else? Regulatory policies and procedures tailored to accommodate the enhanced scientific knowledge, risk-based regulatory approach to recognize the level of scientific understanding, how formulation manufacturing process factors affect the product quality performance, and the capability of process control strategies to prevent or mitigate the producing poor quality products. So we need innovation in manufacturing, because we have been, manufacturing products the same way we have been doing, say, 50 years back, or even maybe 60 years back. So incorporation of quality by design approach based on science and risk assessment principles, and we should look at continuous manufacturing, implementing effective pharmaceutical quality system, say comprising of CAPA system, change management system, management review and of process performance and product, and finally quality risk management. Okay, these are the need of the hour actually. So how all these guidelines or innovation in manufacturing came about in the pharmaceutical industry on a international level? The US FDA, in their internal workshops, when they were brainstorming on various industry aspects, they came to a conclusion that pharma industry has not changed for years. They've been doing the same way. Product is manufactured for several decades. The way they develop products, is the same way. No change. We're not learned from other industries. We're not learned from process industries. And that is leading to failure rate in new drug discovery, which is very high. Attrition, you're aware that attrition rate is very, very high. Even after reaching phase 3 studies, many products are dropped because either they are not safe or they are not, giving any new, or improved treatment options. And drugs are very expensive. Why? Because the failure rate is very high. So a successful drug has to carry the expenses of all the failures as well, isn't it? And not able to meet the unmet medical needs. And therefore, they said we should encourage the early adoption of new technological advances in the industry. FDA wanted to facilitate the application of model quality management techniques in production as well as quality assurance. Encourage implementation of risk-based approach, rather than taking safe way of doing and overdoing things. That happened in the beginning of this century. They started 2001 up to 2004. They were discussing this, and it came out as a, in the form of several guidelines. And for example, ICH came out with Q8, Q9, and Q10. Q8 is pharmaceutical development, and then subsequently they also introduced Q8R, revised version wherein they included the concept of quality by design, including design space. So the three guidelines which are very, very important, which you should be familiar with if you want to follow the quality by design concept for developing new drugs, new products, then you should be familiar with Q8, that is quality by design, pharmaceutical development, ICH Q9, risk assessment or risk management, ICH Q10, that is pharmaceutical quality management. Okay, these three are used together to develop new drugs which are robust in nature. When you have a robust product, there will be less failures, and therefore there will not be product recalls, and patients will not be impacted because of that non-availability of products. So some of the initiatives of FDA and ICH includes process analytical technology, use of PAT in manufacturing, real-time release instead of end product testing and release, QBD for drug products development, risk management, pharmaceutical quality management, and Q11, that is development and manufacture of drug substances. That means even for active pharmaceutical ingredients, there is a guideline in order to encourage industry to use modern concepts and learn from other industries. And of course, lastly, FDA not only expected the industry to change, they also came out with certain regulatory innovations which they followed to approve drugs faster. For example, most of you are aware that new drugs, new chemical entities, or even biological products, they're all approved after phase 3 trial data is submitted to the authorities. You have phase 1, phase 2, and phase 3 studies, and after you submit your NDA along with the data what you have collected in the phase 3 studies, it is reviewed and subsequently it is approved. But regulatory innovation from US FDA was in terms of approving drugs based on phase 2 trials and allowing the company to continue the phase 3 trial. That was one of the major regulatory innovation which US FDA initiated, and under this they have approved several drugs actually, especially in the area of orphan drugs and for diseases for which we don't have treatment today. That is wherever there is unmet medical needs. Many drugs have been approved in the last, say, 10 years based on phase 2 trial and subsequently reviewing the phase 3 data and continuing the approval what has been given to the company. So there were several initiatives which the FDA wanted industry to follow, and they also came out with certain regulatory innovations which they followed and helped the industry. So one of the first thing was PAT, that is process analytical technology. The guidance was introduced in 2004 September. What is PAT? It is considered to be a system for designing, analyzing, and controlling the manufacture through timely measurements. What do we mean by timely measurement? The measurement which is carried out during the manufacturing process, not at the end of manufacturing. Okay, what do you measure? Measure the critical quality and performance attributes of raw and finished materials and processes with the goal of ensuring final product quality. That is, in order to ensure final finished product quality, just don't rely only on end product testing, but introduce techniques for analyzing it during the manufacturing process. That was the basic principle of using process analytical technology. So you have various tools in PAT. So you use in-process analyzers. It can provide non-destructive measurement during manufacturing. Contain information related to biological, physical, chemical properties of the materials being processed. And the analysis can be at 3 different levels. It can be at line, online, or in line. That is, measurement where the sample is removed, isolated, analyzed in close proximity to the process. That means during manufacturing, a sample is removed, isolated, and analyzed. Online is measurement where the sample is diverted from the process and it can be returned to the process stream after it is tested. And inline includes measurement where the sample is not removed, but it is tested online and it can be invasive or non-invasive. Okay, so in many of these cases, there's no wastage of even the material when the testing is done using PAT tools. So why we need to use PAT? The fundamental goal, what is the fundamental goal for a pharmaceutical company? To ensure product quality and the patient gets what he expects. Okay, that is RFT. In the industry we have to be RFT, that is right first time. So PAT helps us in ensuring right first time, because you have process knowledge. Based on the process knowledge, you have controls, and you have in-process testing which is done during the manufacturing, and it helps in improving the product quality and ensures what the patient needs at the end of the day. Yeah. So what do we achieve using PAT? It reduces and eliminates deviations in the manufacturing, improves customer service, that is product availability is ensured on an ongoing basis, because whenever you have a product recall or any product problem, companies discontinue the manufacture of product for some time. So availability of the product becomes a problem to the patients, right? So it helps in improving customer service by ensuring availability, reduces the cycle time, increasing operational efficiency. It reduces the cost because there will be less rework, less resampling, and less retesting because you have all online testing. Then it improves compliance because there will be less deviation reports during the manufacturing, and it improves assurance of product quality. Okay, so reduced need for end product testing is a potential consequence, and it's not a direct goal of PAT strategy actually. Okay, so one of the important things is that you are able to release the product on a real-time basis. So does PAT fit with QBD? That means, do they go together? Yes, because PAT and QBD are like two sides of a coin. PAT forms a crucial component for the foundation of QBD, and it helps generate the process understanding. It provides a practical mechanism for implementing a control strategy for the manufacturing process. It offers perhaps the best mechanism and a regulatory framework for implementing continuous improvement throughout the product life cycle, which is the expectation from the regulators. Implementing QBD without PAT is like navigating a cruise ship, in the icebergs. Okay, so it is possible to survive, but it will be a miracle, if you survive, something like that. So you have to use both PAT and QBD together. So what is quality by design? It is designing and developing formulations and manufacturing process to ensure predefined product quality. That means you have a target, decided by the company, okay, how the product should be, and develop a product to meet that requirement. It's a systematic scientific approach for product development. In QBD approach, we are able to design a product to meet patient requirements. The process is designed to consistently meet product critical quality parameters. The impact of starting material and process parameters on production is well understood. Because if you recollect, the way we develop product, which I mentioned in the beginning of the presentation, in the empirical method, just by taking 1 to say maximum 3 or 5 trials, you feel that you have developed a product. Okay, and there's the end of product development, and you go ahead and launch the product as long as the product meets the specification. So you don't really, are aware fully of various process parameters as well as what impact the starting materials can have on the finished product quality. And critical sources of process variation also are identified and controlled when you use this technique. The process is continually monitored and updated to allow for continuous improvement throughout the life cycle of the product. So what does QBD constitute? First and foremost, we have to define target product quality profile. Okay, that is the performance needed to get clinical benefit and meet customer requirements. That is the main, overall objective of a product development. And then decide and develop a product and manufacturing process to meet this target product quality profile. In the process, identify and control critical raw material attributes, process parameters, and variability. Very, very important. So the process is monitored and adapted to produce consistent quality. Okay, this is what happens when you develop a product based on quality by design. So the application of QBD in pharmaceutical development actually involves multivariate experiments utilizing process analytical technology and other tests to identify CQAs, that is critical quality attributes. And we use risk assessment at every stage in order to identify the right critical attribute for the product. The QBD begins with predefined objectives, as I said earlier, and requires an understanding of the process variables which can influence the product quality. So some of the key components which we need to look at, and some of the terminologies used in QBD, you should be familiar with. Target product profile. Okay, so it actually summarizes the clinical objectives of a dosage form. And for a generic product, normally the target product profile is determined by the key portions of the innovator's product labeling, because generic product is developed based on the innovator's product. The components of TPP for a generic capsule at a tablet dosage form. So you need to look at what should be the dosage form, what should be the strength, what should be the route of administration, whether it's oral, injectable, and what are the proposed indications. These are some of the target product profiles which will list out when you begin, the development process. Then you look at quality target product profile. Okay, I have given one example of, diclofenac injection. How you go about identifying QTPP, in the subsequent slide. Yeah, so QTPP is a prospective summary of quality characteristics of the product, and it is based on target product profile like indications, dose, route of administration, based on which you then decide what should be the dosage form. And QTPP actually lays the foundation for formulation as well as process design. So this is an example what I have given here for the injectable product, wherein how you identify the target product profile and quality target product profile. So for example, here the attribute, what should be the dosage form is the injection, right? And the QTPP is ready to use solution. That is the QTPP. That is because injectable can be in the form of a suspension, it can be in the form of a reconstitute and use, right? Or ready to use. So in this particular case, it's a ready to use injection. Route of administration, IM, IV. For that, QTPP will be whether it's a single use injection, that is in the form of an ampule, and proposed indication, post-operative pain, and QTPP for that will be the product has to be stable, and impurities, it has to meet the ICH requirements. Okay, so this is how for a given product you identify the TPP and the QTPP. Then other components, which we need to consider under QBD during product development is one is target product profile, and we need to design and development of the product, develop the manufacturing process, identify CQA, that is critical quality attribute, and we need to assess and manage the risks involved in the process during the above two steps, and we need to establish what is called as a design space. I'll be defining and explaining about what is design space subsequently. And we also have to define the control strategy. Means what is the strategy you follow when it comes to controls, what you have to put in respect of process controls to manage process variables. So what are the steps involved in QBD? You define the objective and determine what should be the TPP, that is target product profile, determine the CQAs, that is critical quality attributes, carry out risk assessment, and development of experimental design. So design of experiments is used prior to initiating your experiments in respect of product development. Yeah, designing and implementing control strategy, and finally continuous improvement. Continuous improvement is after you actually commercialize the product. Okay, it comes after you develop, you get approval, you commercialize the product, and based on the experience that you gain during manufacture, you have to use it for continuous improvement so that the benefits are benefits reach the patients. So just to summarize what it really means, what does QBD means? Design of product and process to meet patient needs, that is nothing but quality product. What does the patient need? A quality product. Secondly, understanding product and process thoroughly in order to develop a robust product. And third point is continually monitoring the process and facilitate consistent quality, innovation, and of course, based on these things and based on experience you gain through the manufacturing, you go about the continuous improvement programs. So to summarize, QBD elements, I put this in chart just to reiterate what I mentioned earlier. So you have QTPP, that is target product profile to be identified for the product what you want to develop, and identify the CQAs with the help of design of experiments, carry out the experiments, and identify the design space and the control space, put the control strategy in place, and commercialize the product. And after commercialization, go for continuous improvement. So what are critical quality attributes? They are the physical, chemical, biological, or microbiological properties or characteristics. It is affected by critical process parameters and critical material attributes. Okay, that means CQA of the finished formulation will be affected by critical process parameters as well as critical material attributes of the raw material that you are using in the manufacture. It helps us to ensure product quality, safety, and efficacy if you thoroughly identify the right quality attributes. And this is derived from QTPP and prior knowledge about the product what you are developing. So how do we identify critical quality attributes? First, it is through quality risk management. You have to use ICH-Q9 concept. Okay, that is systematic process for identification, assessment, control, communication, and review of risk. And if required, you have to reduce the risk by mitigation plans, and then you go ahead with the identifying the critical quality attributes and developing the product. This here, just as I told you, risk assessment and risk management is very, very important when it comes to developing a product based on quality risk management. So briefly, I'll be touching upon risk management. So what happens in risk management process? You have risk identification. These are the different steps: risk identification, risk analysis, risk control. That means you either mitigate the risk or remove the risk by taking certain steps. And finally, once you reduce the risk, you review the process on a continuous basis and then implement. Okay, this is the, in brief, the risk management cycle. So risk assessment is carried out using suitable risk assessment tools. There are different tools available, like you have flow charts, check sheets, process mapping, cause and effect diagram. These are some of the commonly used tools. And process mapping is quite popular. It is used widely in the industry. For example, development of product specification to ensure bioequivalence limits of acceptable dissolution specification, because linking bioequivalence to dissolution test, this is one of the things what you can do. To do this, you can use the risk assessment process. So cause and effect diagram, which is also called as Ishikawa diagram or fishbone diagram, this is widely used. Okay, so what happens here? You will have a horizontal line. I'll show you the diagram. It will be very clear to you. And you look at 6 different potential causes or categories of causes, like people, machine, environment, methods, material, and measures. Okay, and using these 6 different categories of potential causes, you identify the root cause for a given situation. Okay, so this is the fishbone diagram used in risk assessment. Okay, this alone will take at least 1 or 2 hours to explain. So I just wanted to show you the concept of risk assessment. So I just shown this figure to you. Okay, and the other technique that is used is FMEA. FMEA is a quantitative technique used for doing risk assessment. This is also widely used in the pharmaceutical industry. Okay. So next is control strategy. Okay, control strategy is established to control the material and, for example, to control unit operations, various steps that are involved. For example, if you take tablet manufacturing, you have initial step of mixing, granulation, lubrication, compression, packing, and so on and so forth, right? Different operations will be there. So each of these unit operations, you have to have control strategies in place. And PAT is widely used for setting the control strategy while developing a new product. Okay, and control strategy must be based on process understanding. Okay, very, very important. You should be very thorough about the process what you are going to use, as well as material attributes and process parameters. Continuous improvement over life cycle is the key in the whole concept, and end product testing or meeting specifications unknown cannot assure quality, and therefore you have to rely on PAT to ensure that you have a number of in-process testing carried out during the manufacturing, so that you have real-time data available to release a batch of the product that you are manufacturing. So control strategy can include raw material specification and finished product testing, as well as process controls in process testing. During product development, the influences of process parameters and material attributes on the product quality is thoroughly studied. Only then you will be able to develop what is called as a robust product. Okay, ultimate aim of using this concept of QBD is to develop a robust product, so that you don't have problems when you commercialize the product. So therefore, critical process parameters are very, very important. One of the approaches followed for putting the process control strategy in place is, first you identify and measure the CPP and CQAs, ascertain the sources of variability, because the variability can be there in your raw materials that you are using and the processes that you are using. So you should ascertain the sources of these variabilities and measure limits of acceptable variability. That means to what extent I can accept a particular variability in a given material. For example, you are developing a say tablet dosage form, okay, and the raw material, one of the excipients what you use, one of the important physical parameters which can affect production is say bulk density, right? So bulk density can vary from batch to batch, of an excipient which you buy from a vendor, a supplier, right? So to what extent the bulk density variation can be accepted when you are using the material in your development, has to be decided. That's what I mean by, measure the limits of acceptable variability. Okay, because there will be variability. We cannot have, a material which is not having variability. Of course, it will be there. Okay, so then once you have that, then next is you have to define the critical process steps out of all the because each manufacturer of each of the dosage forms, there will be so many process steps, right? So you have to identify and define which is the critical process step. For example, in case of a tablet dosage form, is wet granulation a critical process step, or is lubrication a critical process step, especially if a drug is incorporated during the lubrication stage when the dose is very low, right? So like that, you identify what is the critical process step. Then you design a process measurement system. That means how to measure, various this variability factors, that has to be in place. Then real-time or near-time monitoring of CQAs, that is through PAT. Okay, and then you develop mathematical relationship between product CQAs and critical material attributes using design of experiments and various statistical tools that you have, and define and design the controls, that is limits, methods, etc. Okay, then you have to implement the controls what you identified and defined to ensure that you have a robust product in place, and then you qualify it and validate the controls. These are the, this is one of the approach that can be followed for process control, putting a process control strategy in place when developing a new product using QBT technique. So I think I put the same thing here in diagrammatically as shown here. So I think I'll skip this. So I mentioned about design space, which is defined in ICH Q8 guideline. So I'll just briefly touch upon that. Design space concept, is that, normally, when we develop a product using empirical method, we look at only one parameter at a time or only one variable at a time. Whereas when we develop using QBT technique, we have multi-dimensional combination and interaction of variables, that is various material attributes. You can use more than one variable at a time and see what is the impact of two different variables on one parameter, what you are looking at. That is the major advantage of using this particular technique. And regulators have allowed us to remain within the design space, and remaining within design space is considered as is not considered as a change. So see, normally what happens in the pharma industry, why industry was not looking at any new ways of developing a product, because once you develop product and commercialize it, if you make any change, a lot of work has to be done to convince the authorities about the change what you have made. That's one of the reasons why they were not looking for new techniques and learning from other industries. So here in the QBD, they have defined what is called as design space, and if you remain within the design space, you don't have to go for any approval from the regulatory authorities post submission of your dossier. Okay, that's one of the major advantages. Normally the design space what is developed during the developmental process of a new drug is proposed by the applicant at the time of submission to the authorities, and then it is approved by the authorities. Once it is approved, then you are free to follow, as long as you remain within the design space. Okay. So I'll be giving an example of, that to make it very clear to you, what I mean by design space. Okay. So design space, in short, it is a multi-dimensional combination and interaction of input variables, material attributes, and process parameters in order to provide assurance of quality for your finished product. And design space is obtained, as I said earlier, experimentally using design of experiments. And as per the regulatory authorities, working within the design space is not considered a change, and this is a major advantage for the industry. Okay. And design of experiments is structured, organized, statistical methods are there for determining the relationship and resulting responses. Okay. So I'll just want to, these are the steps involved in obtaining a design space. Okay. So what you do as a first step in the using QBD concept, you identify the CQAs. That also becomes one of the first step for your design space identification. Then identify the critical material attributes, critical process parameters, then employ risk reduction strategy. That means once you identify this, carry out risk assessment, and this, if there is any risk involved, then you have to go for risk reduction strategy. If there is no risk, you accept it and go ahead with the process. Then you have screening the designs to prioritize risk, and then it leads to our the design space which you use during your regular manufacturing processes. I think this I have covered already. So and here I think some of the examples are given here. If, for example, the target product quality profile, what I was referring to, the physicochemical and pharmacological factors are considered as a TPQP for a pharmaceutical ingredient. Okay, so that is for your API. So in the next few slides, I have, for your better understanding, listed some of the critical quality attributes what we should use during the QBD product development process. Okay, for example, when you of course, when you develop a product, one of the most important input is drug substance. So what are the different quality attributes, critical quality attributes you should be looking at? Description, identification test, impurities, pH, melting point, refractive index, if it is applicable, particle size, bulk density. Okay, and then polymorphic forms, enantiomeric purity, water content, biological activity, permeability, microbiological total count, and if there is a pathogenic count, if it's the total count of yeast and mold, absence of specific objectionable organisms, all these are considered as critical quality attributes. So you have to, out of this list, what is applicable to the product what you are developing, you should identify. When it comes to excipients, again, some are common here, like, you have, of course, in case of excipients, you have to look at concentration, stability, performance of functionality, particle size, bulk density, tap density, hygroscopicity, and microbiological thing is common for both APIs as well as excipients. Then when it comes to drug product, that means you are now deciding what should be the critical quality attributes for the product what you are developing. So what are those things you have to consider? Description, identification, assay, impurities, particle size, polymorphic forms. In case of tablets, whether it is coated or uncoated, capsules with a hard gelatin or soft gelatin, dissolution profile, disintegration, hardness probability, content uniformity, if it's a potent drug with less than 10 milligram per unit dosage form, water content, uniformity of dosage units, pH, and so on and so forth. Okay, these are the different quality attributes what you look at finished product. Some more are there, like reconstitution time, if it is a dry powder which is going to be reconstituted, parent drug products, you have a series of, critical quality attributes like pH, particulate matter, water content, and so on. So if it's a dry powder injectable, and if you use a preservative, antimicrobial preservative content, antioxidant preservative, extractables, that is, if anything leaches from the rubber stopper into the injectable, all these things are considered as critical quality attributes, and you need to identify these well in advance, before you initiate your product development. Then you go for design of experiments, right? I'm not going to details. This is available. Whoever is interested in learning more about this, really details are available. So you use it's all statistical ways of doing it. So in using design of experiments, you are able to use more than 2 parameters, and more than 2 variables can be studied, and you identify the design space based on your study. Okay. So just to give you some couple of example of design space. Okay. See, normally in the conventional method development, we will have a fixed manufacturing process. Okay. For example, say mixing or granulation step for a tablet dosage form. In the submission what I made to the regulatory authorities, I will be specifically mentioning mix for 30 minutes. Okay. Or if it is a lubrication, mix for 5 minutes. That means it is fixed in my submission. Therefore, I need to follow that all the time when I'm manufacturing that particular product. I don't have any flexibility. Okay. What happens in this QBD concept is you study, to such an extent that you know the entire range within which, if you remain, the end product quality is not affected and it meets the requirements. Okay, that is how you identify what is the design space. Design space is the range of a particular parameter or a control. If you remain within that, it will not impact the product quality. Okay, just to give an example, I give an example of mixing time, right? So in the earlier method, I develop a product, keep 30 minutes as the mixing time or granulation time, and I keep doing that irrespective of any variability in the raw material inputs. Because as I said earlier, excipient is one of the main concern for the manufacturers, because it comes from large manufacturers who are not pharmaceutical manufacturers. So the variability will be very high, and based on that, you have no freedom today to vary the manufacturing process controls like mixing time. But in the case of quality by design concept, you are studying a range. Okay, that is, for example, 5 or 10 minutes to 30 minutes you will study, and you will find that if you remain between 10 and 30 minutes, your end product quality is not affected. Then you consider that as the design space. Okay, so you can remain within that, and you can differ it from batch to batch. That's the design space. What is the control space? Within the design space, you make your limits a little bit more stringent and say 15 minutes. You identify 15 minutes as the granulation time. That is the control space. That's what you put in your batch manufacturing record. But depending on the requirement, because there is some variability in the input material, you have the freedom to vary that 15 minutes either to 10 minutes or to 20 or 25 or 30 minutes. So this is the freedom what you are getting as a manufacturer, if you develop your product based on quality by design concept, because you have what is called as design space. That means if you remain within that space, it will not impact the product quality in any way. It meets the requirements. Okay, so this is the major advantage of this particular concept. Okay, So control space is the most stringent one. Okay, so you keep that usually in the production records. That is your batch manufacturing record. But if some problem arises because of variability in the raw material quality in terms of bulk density or tab density, you are at a freedom to increase the mixing time or granulation time to 20 or 25 minutes. Okay, this is the concept of design space and the control space. So there are different types of experiments carried out, using the design of experiments. I've just listed here. I'm not going to the details. You have what is called as comparative experiments, the screening experiments, and response surface experiments. I think I'll skip that because we don't have time for that. Yeah. One of the major objective of using quality by design concept for product development is making a robust product. Only when you make a robust product, there will not be rejection, there will not be product complaints, and there will not be reworks. Okay, that's the overall objective actually. Let's skip this. So we are supposed to, improve the quality of products, processes throughout the life cycle of a product based on the knowledge that we gain. See, earlier, what was happening? Of course, currently, even most of the Indian companies, same thing will be happening because we have not adopted the quality by design concept in India. It is not a requirement. Okay, we continue to use the empirical method for product development. So in this concept, what happens? Based on the product development and technology transfer, I manufacture 3 validation batches. And once the 3 validation batches are manufactured, I'm supposed to use the same process and technique throughout. Okay, I cannot make any changes. If you have to make any change, again, you have to validate that process and then only make a change, right? Whereas in case of your QBD concept, you are expected to improve, ensure continual improvement. How do you do that? As you manufacture more and more batches, you gain more knowledge, right? So you use that knowledge to improve the quality on an ongoing basis. That is the concept of continual improvement, throughout the life cycle of a product. Okay, so that's just to summarizing, the QBD concept here. You have a drug substance and excipients. You have the unit operations to manufacture the product, and you have the process here, and you have various, process, end product testing requirements like assay, uniformity, dissolution, purity, and you apply a PAT, and you always meet the specifications, and the patient is happy. Okay, and regulators also are happy. So this is just try to put that in one slide, the entire concept of QBD concept, of developing a robust product to ensure patient requirements. So next, I think maybe in next 5 to 10 minutes, I will show you 2 or 3 examples, one each for QBD for a generic product, PAT example, and analytical method development. Okay, so in case of say generic product, how I go about using the QBD concept. So I have to identify the innovator and use the innovator's product as a reference product, and identify my QTPP elements and the target. Okay, and for each of that, we need to justify why we have done. That is a process actually. You have to go about in this way. Okay, so for example, dosage form. It's the injectable is the target. Why injectable is the target? Because I have to ensure pharmaceutical equivalence requirement, and it has to be same as the innovator's product. If innovator is having an injectable product, I also have to have injectable product. Then dosage design. It is an aqueous solution. Why? Again, to meet the label claims. Innovator is having a liquid aqueous solution, so I have to develop it. Route of administration, intravenous, again to ensure equivalence. Dosage strength, 20 milligram per ml. Again, it is innovator product is 20 milligram per ml. You go for that. Yeah. Stability, 24 months shelf life at 25 degrees centigrade. Again, it has to be equivalent or better than reference product. Okay, reference product is nothing but the innovator product. Then drug product quality attributes like physical, identification, assay, degradation, sterility, all these things have to be equivalence requirement is there, which has to meet the compendial requirements or the innovator products. Okay, this is how you list out each of the element and justify why you have selected it. Then container closure system. Is it a glass vial or is it an ampule? You have to identify that. So again, depending on the reference substance or reference drug what you have taken, you identify it. That becomes a justification for generic company. Okay. Whereas in case of innovator, they have to scientifically justify why each of these, QTPP elements are identified at the time of product development, because they will not have any reference product, right? They have the first company which is developing the product because it's an innovator product. Okay. Similarly, for a quality attributes for a finished dosage form, how do you go about? Appearance, identification, assay, degradation, all these aspects. In case of a generic company, you identify in such a way that it meets the innovator product specifications. Okay, that's how you go about justifying the critical quality attributes for a finished product. Okay, that's how you go about for a generic product. Now we look at an example of a process analytical technology. Okay, so I'm giving an example of injectable product again, which is a suspension dosage form, sterile aqueous suspension, and objective of the study or objective of this project was improve content uniformity during later stage of filling operation. As you're aware, suspension dosage form, because the drug is not dissolved but it is suspended in aqueous phase, there can be non-uniformity in the distribution of the drug particles within that suspension. If it is not stirred continuously, and if it is not ensured that it is uniformly distributed throughout the suspension. What was found in this particular project was it was perfect throughout the filling process, except the end. Maybe last 2 or 3% of the filling, they had a problem. Okay, so this project was called as RFT and Continuous Improvement Project to provide process changes for improved content uniformity. This was the objective of the project. Okay, now let us look at this. This is how it looks. Okay, it contains a vehicle, water, surfactants, and a preservative. It's a 2 ml vial is used, and 1.2 ml is the fill volume. Okay, drug is 20 to 150 milligram per ml. That is the different strengths are available. Okay, now, so this is a filling operation. You have online turbidity bulk suspension recycle loop here, and sterile filling room, and after filling it comes out, you have plugger. In the same room you put the rubber plugs, then it's capped, and accumulators to take it out, and so in this particular project, offline and at-line NIR analysis of filled vials was proposed. Okay, and that helped in improving the quality of so this is how the graph looks like, when you plot against potency versus approximate percentage filled. If you look at this, how it looks. And so after using this NIR PAT tool, they found that this was the system what was installed actually. Okay, NIR system. So it helped in improving the quality of the last few hundreds of vials which comes out of the filling machine, and ensure a content uniformity of the finished product. Okay, now if you see, widely QBD concept is used only for product development, both for innovator products as well as generic product, but it's also used in developing analytical methods. Okay, analytical methods we can use this concept, and regulators have accepted it. There are several FDA applications made by many companies have been approved by authorities, so it can be used for even analytical methods. Okay, so how do you go about, for example, in method development, you define the method intent, perform the experimental design. The steps are same as what you have for product development. Okay, so now I am giving you example of method development wherein see, normally what happens, you do trial and error again in method development. Okay, depending on the property of the drug being analyzed, you identify the column, and trial and you use one. If it doesn't work, you try another, right? like that. So in case of QBD concept, what you do, you identify the more than one column, okay, right in the beginning. For example, in this particular example, you have CE8 and C18, okay. Buffer also, you identify several buffers, 2, 3, 4, 5, 6, yeah. And mobile phase, you identify different mobile phase right in the beginning, right. And an experimental design comprising of standard set of 2 columns, 5 different pHs, and three different buffers are identified right in the beginning. And so some of the major advantages of using this concept for analytical method also is, one is you get a robust method, because you are doing a large number of experiments in order to arrive at the final method what you are going to validate and then put to use. That's the reason you have a robust method, so there will be less problem during actual analysis, and there will not be any your out of specification incidents and all will be initially introduced as an optional guideline. Almost all the innovator companies currently are using this concept. Generic companies also have started using this concept. Only those companies which are supplying to US, Europe, and other regulated markets, they have started using the concept. But for other markets, companies which are selling only to other markets and domestic market, they are yet to start working on this concept. Okay. And QBD is a scientific, systematic, holistic, proactive approach in product development. Okay. And you get a very robust product, and therefore you have less problem during manufacturing, and there will be less market complaints. Okay. And QBD and PAT go together, and NIR is used widely as a PET tool. I think PET tool. And design space is normally proposed by the applicant. That means the industry, when it develops this product, the industry proposes it to the regulator, and regulatory agency, after reviewing it, accepts it and approves it, and then you are free to follow during manufacturing. But as long as you remain in the design space, it is considered as you are compliant, and there's no approval required again from the authorities. Therefore, this is the degree of flexibility that is given by the regulators to the industry. Flexibility to work within that control design space when you are manufacturing the product. But we need actually consistent implementation of this Q8, 9, 10. It is already implemented in the ICH region, but the rest of the world should also implement it if we have to benefit fully from this particular concept, because most of the other markets, it is not at accepted by the regulators. Okay. Then it is applied in the area of product development, both new drug as well as generic products, and also it is used in analytical method development. I think with that, I come to the end of my presentation, and thank you very much.
Thank you very much, sir, for your detailed presentation on various aspects of quality by design, including various ICH guidelines and the process analytical technologies and tools. I think if we have any questions from the participants.